recap. What did we talk about last time on Friday?
4 types of protein structures. Protein,
Nucleic.
Yep, proteins and nucleotides. Okay, so we finished up proteins, talked all about that, and then we went on to nucleotides
yes.
that make up the big polymers, right? Big giant polymers that make up very important materials in your body, right? Like the blueprint for every single cell of every single part of your body, right? And I'll just put this up there. What is that?
DNA.
DNA. And it's made of, it's got it's a double-stranded helix that's right-handed, right? So it twists like this. And it's got the 2 arms of the ladder and the rungs of the ladder. What are the arms of the ladder made of?
Sugar and phosphate.
Yes, sugar and phosphate. Deoxyribose, good job. You're like, that came out of me? Sure did. Good job, girl. Okay, so deoxyribose, that means the ribose pentose sugar, it has 5 carbons, and it's missing a deoxy. It's missing an OH group, so it's deoxyribose. Sugar phosphate, and it has the phosphate alternating with the sugar. Here, phosphate is in red and yellow, sugar's in gray, all the way around, okay, making those two strands. Now, connecting the rungs of the ladder, okay, are made of what? So we've got sugar phosphate, let's just write sugar plus phosphate alternating, that makes up the backbone, okay? And then we've got, I'll say the rungs of the ladder are made of what? This is key because this is the code, the alphabet of you. Your blueprint comes in these 4 letters. And this is what codes for the alternating crazy sequence all throughout the genome that code for genes and regulatory stuff that's between the genes.
Isn't it the added?
A, T, C, G, Jada said. Okay, so you keep going, Christina. A, T, C, G. What are those though?
They're the adenine, thymine.
That's good?
Yeah.
Yep, adenine, thymine, cytosine, guanine. Those are nitrogen-containing bases. Okay, these are bases, nitrogenous bases, and they make up the rungs of the ladder. And I got some questions in your cards on this. Not so sure the structure of DNA or the bases of DNA. Why do they have to pair up that way? And why is it always A with T and C with G? The overall DNA structure, so that's why I'm going to take a minute to do this. The rungs are the nitrogen-containing bases, nitrogenous bases, and they actually make hydrogen bonds only with the other respective base. A, hydrogen bonds with T, C, hydrogen bonds with G. It is their conformation, it's their structure. They have a single ring or a double ring nitrogen structure. And the way that their nitrogens and their hydrogens line up allows them to either make 2 hydrogen bonds or 3 hydrogen bonds, okay? And they basically can come unzipped. With enzymes or with high temperatures, they can actually, we can take apart the whole DNA molecule. Once it's open, enzymes can sit on there and replicate, copy the DNA. That's what happens before a cell will undergo mitosis that we're going to be talking about very soon, how it replicates, okay? So the nitrogenous bases pair with each other. A goes with T and C goes with G. And the reason I draw 2 lines and 3 is because there's 2 hydrogen bonds that allow these 2 to bond together, and there's 3 hydrogen bonds that allow those to bond together. Okay. DNA is wrapped around proteins. Those proteins are histones, and then those further spiral in on themselves, coil up nice and tight, and they become chromatid or chromosomes. This is the condensed chromosome structure. Before your cells are going to undergo mitosis, like the cells of your skin or of your gut or your bone marrow, and they're doing lots of mitosis, they're doing this constantly. Wrapping up, packing up the DNA, making it nice and tight in the little chromosomes, and then duplicating into 2 daughter cells from that one cell. So this is the extended form of DNA, and then this is the wrapped, packed, condensed form into chromosomes. So the other type of nucleic acid, that was everything we talked about on Friday, pretty much recap. Going into RNA, RNA is really important and it has a lot of different functions. DNA has one main function, and you guys all know that function, right? What's DNA? What's the main function of DNA?
It's like the blueprint of the cell.
Right, it's the blueprint of yourself. It codes for all of the proteins it contains all of your genes for all the proteins of your body, for every single cell, for every organ, for every tissue, everything about you, right? That's not to say the environmental exposures don't impact that and affect expression of your genes and the expression in your body of various things. But it contains the information for all of that. RNA, on the other hand, has a lot of sort of sister aiding responsibilities and roles. It helps in converting that DNA blueprint into actual protein, okay? So this process, I will say, going from DNA, which is in the nucleus, it's contained in the nucleus, we have a big cell, right? The DNA is always in the nucleus. In going from DNA to RNA, and then RNA gets turned into proteins. It's a little whole packaging assembly, it's a factory. Each one of your cells is like a factory, and pretty much its whole goal is to turn DNA into protein, DNA to protein, DNA to protein, and make up all the components of the cell. Replenish and regenerate the cell if it's starting to die, or apoptotically sort of kill itself, for lack of a better word. If a cell is exposed to certain things and it doesn't want to ruin everything around it, it'll sort of decompose in on itself and undergo apoptosis. But in general, this is what a life of a cell does, is to keep you functioning. And cells specialize in different ways, but they all have to go from DNA to RNA to protein. And there's various different types of RNA, but this overall whole process is referred to right here. This is transcription. I'm just going to write the names. So in converting all the enzymes that turn DNA, little pieces of the DNA, the genes, into the message that gets carried out of the nucleus, we call that transcription. And then in turning the RNA message into the key proteins that the genes code for, we call this translation. Translating the message from the DNA into actual protein. And these are destined to either stay in the cell and have a functional role or leave the cell for things like hormones, different peptides, neurotransmitters, things that that cell needs to export and get out to other cells of your body or your brain or whoever we're talking about. Okay, does that make sense? So in this process of going from DNA to RNA to protein, there's a bunch of different types of RNA that help in this process of transcription and translation and getting these key proteins. So some things about the structure, this is more like the function, this is why we care, okay? And then about the structure, RNA is a nucleic acid. It's just ribonucleic acid. Remember DNA, what does DNA stand for? Deoxyribonucleic acid. Same thing, but I'm going to point out ever so slight, little small. There's this one base that we've never seen before, nitrogenous base. Remember we had ATCG? Well, now we have AUCG. Instead of T, there is no T. When your cells convert DNA into message, they use uracil instead of thymine. Okay? So if there's a U in there, you go, oh, there's no way that could be DNA. That's a messenger RNA. That's RNA. That's some type of RNA. And the other component is right here. You see this OH group? Here's the 1 carbon, here's the 2 carbon, here's the 3, here's the 4, and here's the 5. That's a 5-carbon sugar. It's a ring structure. It's got hydroxyl groups coming off of it. It's not a fat, it's a sugar, right? This is a ribose. It's got the OH. So it's not deoxyribose, it's just ribose. Remember, because DNA is lacking this hydroxyl group. So that architectural difference. It has the hydroxyl on this carbon, okay? And it's not lacking it like DNA is. DNA doesn't have that, okay? But it still has the phosphate, sugar, backbone. And it's got the nitrogenous bases that they're bound to through covalent bond. But there's no T, there's uracil in RNA. So there's UACG in RNA. That's a difference. So that's a molecular difference between the two, okay? DNA and RNA molecules are usually single-stranded. So they're not like this. They're not double-stranded. They're not complex. They don't have the entire blueprint of your body. They're like little messages of the genes, and helpers and enzymes and ribosomes and things that make up parts that are going to help get genes into protein. So they got all these little helper roles, okay? But that is structurally how they're different. They're usually single-stranded and they're small. DNA only has one function. RNA has a big diversity of different types of functions, so a wide array of different functions. And if we look at it, here's a few different types. We've got RNA, tRNA, mRNA is the message, like I sort of drew here, when DNA gets read or transcribed into messenger RNA. The mRNA is right here, it's just single-stranded. We have rRNA, that's part of the ribosomes, that helps to, like a typewriter, almost read the message, or a computer, I should say, read the message into actual protein. And so it translates the message of the RNA into the actual protein. So there's lots of different types. You don't have to learn all these different types. Just know there's a lot of different RNA types. They help in converting your genes to protein. So without them, you can't get genes to protein. Like your cell will die, it will not function, you will not be able to survive. All right, so the overall function of RNA is really important, and this is something in biology that we refer to as the central dogma. It's one of these like big entrenched, we've been working on this for 100 years, the central dogma. DNA gets converted into RNA. There's enzymes that literally read along this and read the actual bases and convert that into a message, the messenger RNA. That's only single-stranded. It's a little template that basically says, okay, this is the genetic code right here. This is the message. We have to convert this with all those enzymes and proteins and machinery into globular protein that we actually need to survive. This whole process, DNA to RNA is transcription, RNA to protein is translation. We call this the central dogma, and this is how it works. This is how it works from bacteria, like E. coli, all the way up to every cell in our human body. Does that make sense? Yes? Okay. So this is the DNA, RNA sort of central dogma and why it's really important. We get DNA, we turn it into protein. Now, why can't I just ship the DNA out of the nucleus, go have it read out here by the ribosomes and the enzymes, just clink that into peptides, proteins? Why can't we do that? Tatiana?
DNA is too big.
DNA is too big, DNA is too big, and it's giant strands that are parceled into like individual chromosomes, right? Or even if it's extended, it's in there, it's way too big. And it's very precious, right? It's very precious. If your DNA leaves even through, there's little pores in the nuclear envelope. If it leaves out through there and something happens to it, it starts to break down or degrade, or it leaves out of the cell, that cell has no blueprint. It's gone. It can no longer function. It's going to cease to be able to perform everything it needs to perform. So when something is very valuable, we make copies of that, and we keep that precious commodity in the vault, right? We make copies, we export those copies, we say, hey, we need this one right now, make all those. Okay, now we need this one right now, so we're gonna export the message, okay, make all those. So it's really, if something happens to a cell's DNA and it starts to leave the nucleus or get ruptured and falls apart, that cell will sort of kill itself. It'll undergo apoptosis because it's no longer contained and in the right, proper way that it needs to have its DNA. All right, so another important nucleotide is ATP. And I know, raise your hand if you've heard of ATP. ATP, okay, we should come up with a little song or a little rap for ATP. I did a lot of that when I was growing up because it helped me like remember some of these crazy biology things. ATP is actually considered a nucleotide, okay? Just like we talked about with DNA and RNA. So it has similar structure with the sugar phosphate. So it's adenine, okay, that nitrogen space, plus ribose, plus 3 phosphate groups, P with O. Okay, so it's got, we call its overall name is adenosine triphosphate because it's got 3 phosphate groups attached to the end of it. So you think, okay, who cares ATP? What's the significance of ATP? Why do we care about ATP? It's used for energy. Yes, used for energy. And powers almost every single thing that you do. Almost every single thing that you do. You can come up with a list of different things. You're using ATP energy for every single one of those things. The vast majority of what you use chemically in your body, why you need to eat, why we need to have different nutrients of all the different macromolecules that we've talked about, is so that we can generate enough energy to power our metabolism, our basal resting metabolism, and power our ability to do all the things we need to do. Physical activity, to think, to move, to behave, to understand, to speak, to just sitting there and thinking, you're using ATP. You're using energy. Those neurons are using in their pathways and connecting and talking to each other, and that is using ATP. So it's the biggest energy currency that your body has. You have some other little high energy sources, but almost everything ultimately leads to high levels of ATP. You burn like 1 billion molecules of ATP a second. Some ridiculous number. Now, if you're an athlete, if you're an Olympic athlete, that number goes even higher. Okay? So it's really, really important. Here's the 3 phosphate groups. Here's again the ribose, the 5-carbon little sugar, cute little sugar, and the adenine, the nitrogen space. It's got nitrogens in it. It's got 2 rings. That's the nucleotide structure. That's ATP. This one has special importance because it's the energy currency of all cells. And again, I'm talking through evolutionary times from bacteria to women and men, right? This is the energy currency. I'm sorry, I feel like I'm always right in your way. I have to step aside, I have to dance. Okay, so high energy molecule used to make macromolecules, all the ones we've talked about. Even making DNA and RNA, you need ATP. You need ATP to actually fuse all of those nucleotides together to make nucleic acids. Okay, so take it back to the beginning, and we learned about hydrolysis and dehydration reactions. Those take ATP. So they actually need ATP and the proper enzyme to go, whoosh, okay, let's put these molecules together. Let's form a new polymer. Forming a new polymer takes ATP. It takes energy currency in the form of ATP. So it makes those big macromolecules we've been talking about. It powers reactions, a lot of different processes, metabolism of the body, muscle contractions, nerve impulses, all of that. I do little application questions sometimes in my bio courses, and I say, there's a molecule or a drug that you ingest, and it irreversibly binds to all your ATP. So it binds onto the end right here and makes it so you can't break this molecule down. It's just floating around, it's everywhere, but it can't be broken down. You would die, actually. Everything that you do would cease to operate because that's how much you're relying on breaking the bonds of ATP to do work and to do all these different things. So basically, you've got the adenosine with the triphosphate group. What you can do is with water, you can break off that terminal phosphate. And in doing this is a high energy chemical bonds. So when you break off this third from the second phosphate right here, you break that off, it provides energy. So usually that instills a conformational change or an enzyme to change position. Remember we talked about proteins and how important it is that lock and key fit? You can use that energy to that next molecule that it's transferring the energy to, just by cleaving off that terminal high energy phosphate group. And this provides the energy to do all those things that we've been talking about. So it's very, very important. And it can be, oops, where did it go? Well, I have no idea. It's just completely gone. It'll come back, maybe. There it is. I have no idea what it got out of it. It got into the wrong place. Let's see. Right there. It's basically the ATP cycle, and we'll talk a little bit more about this later. Come back. There we go. Okay. Don't know why I did that. It's basically a cycle. Your body can also, when you have enough energy and you've consumed enough nutrients and you have fuel, your body can actually put the phosphate, your cells, your enzymes can put the phosphate back on and turn ADP, adenosine diphosphate, back into adenosine triphosphate by anchoring on that terminal phosphate again. In other words, you can recycle your ATP. It's a revolving door, right? Energy reactions that give energy will help that ATP glue back up together so you can use it again. Reactions that need energy, that need to be energy put into them, will actually use the ATP and break it down. So you're constantly, it's this revolving door of energy currency that all of your cells and parts of your body are dependent on for surviving and being able to do work or do anything. Okay, so that's the end of this one. Any questions on ATP? That was the last section of our nucleotides, or macromolecules of nucleic acids. Yes, Jackie?
So when how people start really working out and they get more strict, does your ATP and metabolism then suddenly go up higher?
Yes.
Okay.
Yes, it speeds up. And that actually really gets stimulated in several places, especially the skeletal muscles. So when you're doing a training program, your muscles are one of the first places that go, oh, whoa, we're getting a little bit more exertion, we're getting a little overload. Okay, we need some more ATP, we need to burn a little faster, we need to speed up metabolism. So they start doing all the enzymatic reactions that go into aerobic respiration to speed up. So now they're speeding up, so they're using more ATP. And actually your muscles are actually super amazing. You have mitochondria in all your cells. We're going to get to that pretty soon. Mitochondria are the little powerhouses, the little organelle that's inside of your cells. And they allow the production of massive amounts of ATP. We're going to look at mitochondria just right now, actually, so this is a perfect segue, Jackie. So the ATP is made in the mitochondria. When you're doing a new workout, whether it's load-bearing, strength training, or stamina, like running, your muscles get that stimulus. Ooh, we need to crank out more ATP. We need to increase metabolic rate. Your metabolism will actually increase. It will speed up. It'll cause you to want to eat more. So you'll eat more so that you can sustain that. Your muscles will actually start, the mitochondria within them will get the trigger to start dividing. So within your individual muscle fibers, the mitochondria will start pinching. They'll start proliferating and budding. You'll have more mitochondria in the individual muscle cells. You have more mitochondria. Mitochondria are the main powerhouses. I call them the PGE. I hate the PGE, but PG&E, but that's like what they're making all the electricity, the energy, right? So if you have more mitochondria, each muscle per fiber is making out way more ATP, right? So more energy, so all the cells and tissues of your body can use it when they're doing more physical activity. That in itself helps to reset or increase your metabolism, your metabolic rate. Yes, so absolutely it goes hand in hand. Believe it or not, even eating burns calories. Even eating, like, increases your metabolic rate because it takes activity to sort of do that, eating and thermogenesis, keeping yourself warm. Okay, so switching ahead, we're going to metabolism and nutrition. So exactly that topic of how we burn and how we use our muscles, and we do all these metabolic reactions. This one is called Metabolism, Nutrition, and Health overall. We've been talking about this already, kind of woven throughout. So I can't teach about fats and sugars and proteins and DNA without talking about the body and nutrition. So you've already gotten little bits of this all throughout, and hopefully this is going to intertwine some of these things together. I will warn you. The first about a dozen slides, is very biochemical in nature. It's got a lot of sort of chemistry and biology. Is sort of understanding aerobic respiration or metabolism. This is a picture actually of my student, McKenna. She was a Kines HHP student. She took some beautiful pictures in lab. This is actually the thyroid, the human thyroid gland. Do you know where the thyroid is located?
Yeah.
I have a stiff one right now. It's right up in here. It's like a little butterfly gland that wraps right around your laryngeal cricoid cartilage. It's right there, okay? And it actually is sacs, these follicular sacs, that helps to make thyroid hormones, T3 and T4. Those actually help set metabolic rate or metabolism. If those are low, your metabolic rate is going to be lower. If you have higher quantities of those, you're going to have a higher metabolic rate. In general, though, they just help to keep it in balance. They help to keep your overall metabolism in balance. And we'll talk more about the thyroid and thyroid hormones when we get to the endocrine system, because it's an endocrine gland. And it's a hormone that helps to regulate that. So we're just not going to mention too much about that now. So overall metabolism, we'll do an introduction to metabolism and then go into nutrition. All right, now that's my daughter Lola. She was being pushed by a dolphin. We go to Mexico, we go to the Caribbean in Mexico, where I told you I take my Janterm students to study sea turtles. Take my family there, that's how I first found out about it. And she had little dolphins pushing her feet. So you have to make yourself real stiff and they like push your feet. It's so cute. They were just adorable. So metabolism basics. In aerobically respiring creatures like us, aerobically respiring means we take in oxygen and we exhale CO2, okay? We take in oxygen, we need oxygen to survive. We can't do any metabolic anything or breaking down or building anything unless we have oxygen. So the overall metabolism reaction is, and I make it real simple, this is just real, it's way more complicated. There's all these things that are feeding into this and happening, but the overall basics are oxygen plus food stuff, nutrients, goes to CO2, ATP, water, and heat. That's it. That's the overall metabolic reaction or flow that happens. You need to take these in, and then these are the output or the product of that. And that's my little guy Pee-wee back when he was eating corn. He could dust like 5 corn on the cobs, just eating, eating, eating. Metabolism is all the chemical reactions that occur in a cell and in a person. So it's all these metabolic reactions that allow these substrates to go to these products and give off ultimately energy and CO2. So metabolic pathways and enzymes are really important. So as part of this process, we have to understand what an enzyme is and what a metabolic pathway is. If you've noticed too in my slides for study guide, checklist, stuff that I'm going to help you with, when I put stuff in bold or italics or a different color, you need to know that definition, you need to know that term, right? There's a reason I'm like, yellow, right? Pay attention to this. So I try to highlight those all throughout all my lecture slides. And I put a lot of time into my lecture slides. So they're the things that I really want you to know. Most reactions are linked in a chain or a web, and we call that a metabolic pathway. Believe it or not, you guys are learning a little bit of biochemistry with this. This is a little bit of bio and a little bit of biochemistry. So a metabolic pathway is a series of reactions that are linked together, like A goes to B, goes to C, goes to D. These begin with an initial substrate, and they end with the final product. And there's a lot of enzymes along the way. The enzymes, what are enzymes made of? What are enzymes made of? We talked about like antibodies, and we talked about enzymes. In one of the macromolecule sections, proteins, they're proteins, they're a type of protein. Remember we went over circ, the structural stuff, the enzyme stuff, I gave you that acronym. Almost all the things of your body are made of proteins. So enzymes are made of proteins, and they help convert one substrate or two substrates into products. Okay, they catalyze or make those reactions speed up. So here's an example of a metabolic pathway. These are examples of enzymes that are converting substrate, which is the starting material, into product. And in this case, one product is another enzyme substrate, right? And then this product is another enzyme substrate. So it's a series of reactions, they're all kind of married together to give this final product here labeled as F. And this is usually something that is really important for your metabolism and your overall processing in your body. So we've got A to B, different specific enzymes help to catalyze those reactions, and you end with a final product or more final products. So enzymes are really key for this process for metabolic pathways. Here's an example of an enzyme. Kind of looks like a Pac-Man. We always kind of draw them this way. They're like a Pac-Man mouth. They've got an active site. That is an active location where the substrate or substrates can fit in, okay? And they fit in, and there's usually a protein. Remember we talked about the levels of organization of proteins, right? There's a protein structural change that happens. We call it a conformational change. That when that enzyme binds its key substrates, it changes conformation a little bit. I like to call it, it's called an induced fit, but it almost is like the enzyme is giving a hug to its substrates. It's like, come on in here. You know, people give you a hug like this. The enzymes don't really do that. They get in and they like grip in there. They give a hug, they give a proper hug. That's how I like to think of it, okay? So it's like they're hugging kind of. They bring in, they change their conformation. What that does is it brings the two substrates in a relationship where they're real close together. Bonds are typically broken, new ones are formed. So you start with two substrates and you end with two totally different products, okay? So enzymes catalyze this. This is like probably 10 lectures of enzymes and enzyme kinetics in a biochem or biology class, but this is all we're gonna do in here, okay? So they are magic though, they're little magicians. They just speed up reactions. They bring things in close proximity. They allow a reaction bonds to break and form, and they create something new. So really they speed these reactions up. Because these guys are all just diffusing and floating around, but they help to speed that up. So they're metabolic assistants. They speed up the rate of a chemical reaction. Now, does it mean they make something happen that wouldn't normally happen? Not really. These reactions might happen, but they might take 100 years or 100 days, right? An enzyme is there in that environment, in the cell, in the tissue, allowing it to just get what it needs, put them in close proximity, speed up that reaction that might take a lot longer, okay? So they help convert substrates into products as the take home. They have an active site, the Pac-Man mouth, that's the active site. Active sites are all different, but the point is they're made to fit really nice with whatever the substrate is that they prefer. And there's thousands of different enzymes in your bodies, tons, and they all do a myriad of all these different reactions. So they break down a substrate into new products or combine substrates into a brand new product, okay? And notice when the product is formed and it diffuses out, it's released, the enzyme goes back to its normal protein conformation. That means it's reusable. It can be recycled and used again. Okay, it's ready to do the reaction again. Does the whole thing, again, opens up, does the whole thing again, opens up. So you can use these over and over again. They get reused and recycled. This is showing the same thing from your textbook. The enzyme can take in a substrate, ultimately convert it to products, right? Or take in a couple different substrates, put them together in a certain way so where you have a new product. Same thing here. This is an example of an enzyme sucrase. Sucrose is the typical sugar that's in like table sugar, right? So like sucrose, it's in brown sugar, it's in white leaf sugar, it's in all that. This enzyme actually brings together glucose and fructose sugar molecules, monosaccharides that we've already talked about, right? Monosaccharides, brings them together because it has an active site that fits both of them and actually makes a new glycosidic or bond, covalent bond between those. So now ultimately, I'm going the other way. Substrate takes in this case, sucrose, and then it can actually break down sucrose and bring it into its two respective monomers, which is fructose and glucose, freeing those two separate monomers. So and some of these enzymes go the reverse. So this sucrase is actually taking in sucrose, table sugar, splitting it with water, and now you have individual fructose and individual glucose. What's that reaction called right here? Right, hydrolysis. This is an example of an enzyme that is doing hydrolysis to break down something like sucrose when you have brown sugar in your coffee. This is the enzyme in your gut, in your cells of your gut, that are actually catalyzing this reaction. Now you have free glucose and fructose, and you can aerobically break those down. So you can break those down in metabolic pathways in your cells, and we're going to look at what that looks like.
Is the glucose and fructose the final product?
In this case, the two products would be glucose and fructose.
Okay.
Yes. So I started with these. I should have started up at the top. But yeah, there's enzymes that go the other way as well. They sort of build. This one is breaking down. So this one is catabolically breaking down sucrose into its two monosaccharides. Here's an image of an actual rendering of an enzyme with its active site and the substrate and how it fits in there, kind of fits in and hugs it and induces its change in conformation and mediates the reaction, catalyzes the reaction to go. Okay, so how are we doing on time? Just want to make sure.
10 minutes.
Okay, so we'll get through the difficult. This is sort of the difficult part, and you don't have to remember every single thing here. So I'm going to highlight what I started to want you to know and keep track of, because I realize this gets into metabolism is very biochemical. Like there's whole biochemistry classes that do nothing but metabolic reactions, and we're going to do it in just a few minutes. Okay. So the point is, the take-homes are cellular respiration for us who are aerobic organisms. We need oxygen. Oxygen and glucose are transported by your blood to all the cells of your body, okay? So we have to break down glucose in cells to get the energy from those monosaccharides, especially glucose, that's our preferred sort of fuel source,